to biotechnology. Today we're going to do an experiment or a demonstration of converting bacteria, in this case E. coli bacteria that I've grown on this plate here, into another form where they make green fluorescent protein. This is a standard experiment sold by Biorad, and you can look at their instructions online and other videos of people doing it. They don't make as many mistakes as I do. So what I'm going to do is put this transformation solution, which is a buffer that also contains a lot of calcium ions in it. The calcium ions connect to the cell wall, and they also connect to the DNA plasmid that we're going to add to help it to enter the cells. So we're going to take a measured amount of transformation solution and put it into these two little centrifuge tubes. The green one I'm going to put green fluorescent protein plasmid in, and the other one I'm not. It's the control, so that we can see that something has happened, and so that we can see that it is because of the plasmid and not because of something else. As a recap, a plasmid is a circular piece of DNA that is not the main information that the bacterium needs to grow itself. It's an extra piece of DNA. This is the bit that they often switch out with other microorganisms, and it's the one that we're going to use in biotechnology. The one we're going to use contains an arabinose receptor, so it switches on and off in the presence of arabinose, which is a type of sugar. When it's on, the part that would normally break down arabinose will be activated, but that's been removed and replaced with the sequence for green fluorescent protein. So our bacteria, if it takes in this plasmid, will make green fluorescent protein in the presence of arabinose. On the other side of the plasmid, there's an always-on section. There's a piece of DNA that contains an ampicillin resistance gene called BLA, beta-lactamase, and it is not got a promoter, unlike the green fluorescent protein part, so it doesn't make a lot, but it only takes a small amount. So now I'm going to collect over here, switching back to what's actually going on. I'm going to collect a colony from the plate. You can't see it because they're very small. This might be a mistake that I made, as we will see in a moment. Not a moment, it's a long time. So that's going into the first centrifuge tube. That's the green one. It doesn't matter at the moment if they're mixed together because they're both identical. They've both got transformation solution, which has got calcium plus, 2+ in it, and buffer, which will help them to avoid turning acidic or turning basic if some of the bacteria die. They've both now got bacteria in, which I've scraped off this plate, and they should both be genetically identical because they've come from colonies grown from a pure collection that I bought. So they should be fine. Because they're small, you're supposed to use one colony because then you know that it comes from one cell. Because they are very small in this case, I've used slightly more than one. It turned out not to be enough. So I'm going to put the lid back on the plate, and then I'm going to add a tiny amount of the green fluorescent protein plasmid, which is a chemical, a piece of DNA. And it, oops, okay, well I switched over, oh, there, okay. So I'm cooling it down for 10 minutes in icy water to get it nice and cold. And then I will put the plasmid DNA in there. So that is now nice and cold. And we will put the plasmid DNA in. Now I might do it off screen. Some of the time it's hard to see. It's only a tiny amount of DNA is required because obviously the cells, every cell needs one molecule of it. So it's only very little. Most of the bacteria will not be transformed by this process. It's not very efficient. And so we will need to get rid of a lot of them in the intermediate step, but at the moment we're trying to transform them, which means the DNA is going to get from the outside of the cells to the inside. At the moment I'm getting them to a standard temperature, which is 0, by putting them on this ice and adding the plasmid DNA to just the green centrifuge tube. The next step will be to put them into a water bath at a high temperature, a temperature that's enough to kill them for only a short time. It will be 50 seconds. That will take a while to get through the walls of the container, so the ones on the outside will probably die and the ones in the middle will probably not die, and somewhere there will be a perfect temperature where they're stressed by the heat but not stressed enough to kill them. Those ones we are hoping will survive, and they will then absorb the plasmid. One of the theories about that is that the plasmid sticks to the cell wall by electrostatic attraction, the problem being that the plasmid or DNA is negatively charged, as we know from gel electrophoresis, and so is the cell wall, which means they would repel each other, so we need a doubly charged calcium ion to sit in between and stick them together. When they get exposed to the heat, it is theorized that parts of the cell wall flip over and that puts some of the plasmid on the inside, but it's unclear. It might also just break some proteins and allow a little bit of the DNA inside. It doesn't take much, so now I'm going to go straight from the ice into the hot water bath and then back into the ice again, and that should do the trick, but you can't see anything at this point because the bacteria are in there. There are a very large number of bacteria in there, and this is one of the reasons that biotechnology is commonly done with bacteria, because because there are a lot of them, it's easy to make a lot of bacteria, and so if nearly all of them, so 999 out of 1,000 die, that is not a big deal. A new colony will grow from a single bacterium in a day or two. The drawback of this process is obviously that the heating and cooling system is fairly brutal and very statistical, so most of the bacteria will die, and it's very inefficient. The good thing about bacteria is that obviously once we've got some that do the thing that we want, we can grow a lot. So we only really need a few to work, and then we can make as many as we like. I've put it back on ice now to cool back down again. That saves them from dying, gets them back down to normal temperature. They gradually cool back down to ice temperature really, but I don't need to leave it for that long. I just need to get them back down to room temperature so that they can then start to grow. I will have to feed them then so that they can divide and start making antibiotic resistant chemicals, so that when they get plated out onto the Petri dishes that we saw earlier, they will not be murdered immediately by the ampicillin in the Petri dishes, because obviously that would be bad. So now they've been standing around for a little bit, and it's time to feed them with a little bit of nutrient broth. So this is just food for them, which is amino acids and sugar. It's a bit of salt and buffer. There is a standard solution, or one of a variety of standard solutions. The amino acids are made by breaking down some protein source, typically vegetable or animal protein source, often yeast, and that divides it into pretty much into the individual amino acids, and you can use it to feed the bacteria with. Also got glucose in there as an easy energy source. So I'm just going to add a standard amount to each of these that will recover the buffer capacity and also feed them, hopefully get them growing and making beta-lactamase, which is the antibiotic resistance thing. You can see that I've got various plates. I've got the ones with the PGlo plasmid and ones without for reference. And I've got a plate with ampicillin and a plate with ampicillin and sugar. I keep saying sugar because I keep forgetting the name of it. So fast forwarding through a few minutes of time where it's just waiting for the bacteria to grow, I'm going to then go on to spreading the bacteria over these plates, which I'm going to do by taking a known quantity of each solution. So I should have about the same amount of bacteria in each, and spread it over these plates using an inoculation loop, which is sterile. I'm not going to use the same one for the ones with PGlo and without. I'm also going to start with the ones without, because those are the ones that I'm expecting to die, and I don't want to give them a chance of survival by putting some in by accident, cross-contaminating it. So I'm going to put the same amount into each one, and then I'm going to spread it out as evenly as I can. This is a little bit fiddly. If you use a normal inoculation loop that's metal, it's very easy to scratch your gel. These ones are plastic, they're slightly easier, but I still put some lines in it because I'm not very good at it. So I'm just spreading it out over the surface. This happens a lot because I'm going to spread all of these out. I can use the same loop for all of the ones in the same thing. I should, however, not have done it in that order. Oh yes, that's okay. So I did it right. I started off with the nutrient broth one, then I went to the one with an additive, and then I went to the one with 2 additives, because obviously we don't want to get the antibiotic into the one with no antibiotic in, because then there's a good chance that we will see nothing at all. While I do the rest of them at a slightly sped up rate, I can explain to you what I expect to happen. So I expect on the control, on the without plasmid, I expect on the LB, the food one, I expect some colonies to grow. There should be a lot of colonies, in fact, because the bacteria shouldn't die that much from the heat. On the antibiotic-containing one of both types, I expect no colonies to grow at all, because there is no reason for them to be antibiotic resistant. There is a small possibility that they could have gained an antibiotic resistance gene from the wild, and if I leave them lying around long enough in the lab, they will pick up an antibiotic resistant strain of something, or a fungus, which won't be affected by the antibiotic. So eventually something will grow, but it'll take a very long time, and it will probably grow very slowly. In the PGlo version, I would expect that the antibiotic containing plates, that's the 2 at the bottom, that they will also contain bacteria, but far, far less than on the blank plate, which will contain bacteria that have not taken the plasmid and bacteria that have taken it. We would imagine that in the top one, in the LB plate, it will be about the same as in the LB plate of the other one, but because I didn't carefully take exactly the same amount of bacteria in each, it won't be identical. In the 2 at the bottom, we would expect them to grow about the same, because we wouldn't expect them to grow about the same, because when the green fluorescent protein gene is turned on, we would expect that to reduce the rate of growth of the bacteria. So we'd expect the colonies to be smaller, but they will make green fluorescent protein. And in the one without the sugar, then we would expect the colonies to be the same number, but to be bigger. That probably won't happen in this case, because in my experience, the green fluorescent protein doesn't slow the bacteria down enough for us to see a difference. But it does matter in other things, such as when we make hamster cells make human proteins, or when we want to make bacteria make spider silk, then the product that we're trying to make is slightly poisonous for the cells, so it either eventually kills them or it severely hampers their growth. And in an example that I used for my for an exam question, growing human proteins, sometimes we use a promoter that's so powerful the cell doesn't do anything apart from make the protein, and then obviously they're not going to divide when that gene is switched on, because they just don't have enough amino acids to do that. That is not the case here, so we won't see a very big effect. 2 days later, and I tested it, and nothing grew on any of the plates apart from the LB plates, which suggests that the transformation hasn't worked. So now we need to analyze that, and I also need to do it again. So what's happened? The transformation hasn't worked because none of the bacteria have survived, which means that probably the antibiotic has murdered all of our bacteria, well, almost certainly, because on both of the LB plates we've got colonies. Luckily, we can see now that there are better colonies on here. Here they are, lovely colonies. So that is exactly what we were wanting to see the first time and we didn't see. And I'm going to be more careful with all of the other things as well to try to identify what was going wrong. So I'm going to take one colony from each and put it in each tube. I might even overdo it this time. I can't remember what I did. But now I can take a proper colony, a proper round colony. E. coli are white colonies and they have a certain smell, so I know that they're E. coli. I'm going to use green and orange. I'm putting 2 colonies in, I see, because I'm a bit worried about it working. And I'm going to repeat the whole thing. I'm also going to be more careful about the plasmid, because I'm a little bit worried that my plasmid might not have worked. Another possibility is that the ice, it does say in the instructions to use crushed ice, and I didn't have any. So there is a possibility that the ice wasn't cold enough, it wasn't in contact with the tubes enough, and so I'm going to use that as well to try to make it work this time. So testing my plasmid, I would like to see, these are my ultraviolet torches, would like to check and demonstrate that it is not green and fluorescent, because that would be a possibility. You can see it's purple, which is not fluorescent. It would be fluorescent if it was converting my purple light into green light, which it obviously isn't. So I'm going to take that. You can see I'm wearing one glove and not wearing a glove in the other hand. That's practically useful, because then you know which hand is dirty and which hand is more dirty for bacteria and which hand is dirty for writing. I'm going to add a little bit of transformation solution to my plasmid. It's still not fluorescent. There is a little bit of green from the paper that generates a fluorescence. The inside of the label you can see, but the actual liquid is not fluorescent. I'm going to then add that to my tubes and do the experiment. I was a little bit nervous about my PGlo plasmid at this point, because I thought it had frozen. I kept it frozen and it probably shouldn't have been. It said on the pack, don't do that. So I'm not quite sure about it at this point. However, I'm going to go through the motions. I'm going to put it in proper crushed ice this time to make sure that that isn't a problem. I'm going to do the time as well as I can, which I was sure that I did correctly last time, but I'm going to do it again well. I'm using the same containers that came with the kit, because the shape of the containers will affect the heating and cooling. So I'm not going to use any different centrifuge tubes than were with the instructions, because obviously the 50 seconds apply to that size container and no other size. I'm not sure what's going on here, but then I am going to feed them and give them plenty of time. I'm probably going to overdo the time on the feeding. So at the moment I think it's in there for its time. It's probably nearly done. It's 45 seconds or something. I'm just making some holes for it, and I'm going to poke it straight back in. So now it's going to cool back down again for 2 minutes, and then it's going to go back to warm back up on the bench. So my 2 minutes are up. I jumped over that. We're going to go back over to the bench, add some food, which is our LB. I think that my LB is still okay. It certainly isn't contaminated with anything, because then in my other Petri dishes, other things would grow, and it was all E. coli. So I'm reasonably sure that that's correct. I'm going to add this and wait again to let them grow, but I'll wait a little bit longer to try to be sure this time. Zooming through that at a high rate of speed, we can hopefully manage to catch the time. Now I'm mixing it, letting it sit for a while. It's all, you can see by the time going fast, it's not real time. I'm then going to plate them out just as before. Here we go. I'm hoping we slow back down to normal time. There we go, back to normal time, doing exactly the same thing as before. I've got fresh plates this time so that we can start from new. Luckily, I made a whole classroom full, so there were plenty. So now I'm going to do exactly the same. Add a measured amount, spread it about over the plate. You get the drift. It's exactly the same as before. Going to let it soak in. Going to leave it for nominally overnight, and then we will come back and see what grew. But obviously there's going to be a lot of this plating out because I've got another 5 of these to do with all the other alternatives. So here's me in sped up mode doing all of these. It's exactly the same as before. Put some on the Petri dish, spread it around with a inoculation loop, trying to keep the dirty hand away from the bacteria and the bacteria hand away from any writing implements and other things that I might want to touch. Also the camera. That's one of the problems with doing this kind of work. I need to touch the camera and the bacteria. So now I've spread it all out, and there will be a cut to black where time passes. This is not actually 1 day later, it's several days later, because it turned out that it appeared to not work. I did it yet a 3rd time with more bacteria like this from one of these, but over time they grew on the original plates and it was fine. So it turns out that probably my broth, which isn't LB, it's an equivalent, is not as fast as the one that I was supposed to use, and so the times didn't fit. But that was in addition to it not working the first time at all, which was probably due to the temperature and possibly the amount of the plasmid that I added. So these are the colonies all in there. That would be LBA. So these are with the antibiotic, and you can see there are plenty of colonies on there, and they've grown fine. That's one without the plasmid, and you can see nothing's in there. That's one without the plasmid and with antibiotic, and you see again nothing's grown there. Getting our ultraviolet light out, we can see what's gone on, and we can see several important things about this, I'm hoping. It's taking me longer to get that. There is my violet to ultraviolet light. You can see it. The camera sees it better than you can with your eyes, but we can only see the camera here. Let's try and hopefully do it in shot and not completely out of shot. So those are the two that I want to look at. LBA without any arabinose in there, so they shouldn't be making green fluorescent protein. You can see the gel fluoresces green slightly, but the colonies look darker, so they're not doing anything at all. Those round colonies on the top are what we're actually looking at. The gel is fluorescing slightly in this wavelength. But we will see the difference when we get to the other one, where the detector on my camera gets overloaded and it has to auto correct. There we go. Boy, it's so bright, and it's so bright that when the camera corrects for it, the green from the gel goes dark. So if I bring the other one back close into shot, into the same beam, you can see it's definitely not doing the same thing. Those colonies are green and fluorescent, and the other ones are not. There is another thing that I want to show you. I think I'm going to have to show you in stills because I didn't take a video of it. So we'll do that. Oh, hang on. No, this is just a copy of the same thing. This is obviously some that have grown more. So I put more on this one. This was the third attempt. I put more bacteria in, and you can see it's kind of full of bacteria, the one with the ampicillin and sugar on the right, and again it's bright green, and the other one is near purple. So they're doing exactly what you would expect. So the other thing I wanted to show you that usually doesn't happen is this here, where some of the colonies glow green very brightly, and some of them are also green, but they're not as bright. So even though I'm moving the beam around here, these are all stills, but I'm moving the beam around, you can see that some of them are definitely brighter than others. Some patches are probably far brighter than others, but it's hard to tell exactly how much. And that is because there is no requirement for a bacterium to only take up one plasmid. And so there's a probability that some bacteria will get more than one, and those will obviously be favored in this antibiotic-rich milieu, and so they will survive better, but also they will make more green fluorescent protein. It may not be double as much, but it will certainly be more, and that will show in the results. If we want to make bacteria have more than one plasmid, however, it's difficult because obviously they only need one copy of the antibiotic resistance gene to actually survive here, so they will eventually lose the second copy of the plasmid as they divide. There are tricks to solve that, and usually it's done by using multiple antibiotic resistances, so that you can have several plasmids for the same thing, or of course you can make a stronger promoter to make more using just one. So that is the practical for this time. I hope it was interesting to you. You can see all of the instructions on the Biorad website. It's under PGlo. They are free to everybody, and you can look on YouTube as well. There are several other groups who do exactly the same thing and take videos, and obviously they don't make as many mistakes as I did, although making mistakes is sometimes useful to learn things. See you next time.